Thank you so much, Nella. And I really want to thank the organizers for this wonderful opportunity. It's a great series. I've taken a look myself at some of the other talks that have been in the series, and I think it's amazing. And it gives me the pleasure today to continue the series on microbes because I had a look and realized that you've done a lot of microbes in the last couple of weeks, which is really great. So for the session today, one of the things I really wanted to do, obviously you're going to ask questions, but I also want to ask you questions along the way. And the whole idea of doing this is just to see how much you have followed your previous sessions and how much you're following in this session. So when we say microbes, what are they? Usually, they are living things too small to be seen with the naked eyes, right? Now, the question is, they can be bacteria, they could be viruses, they can be fungi, they can be protozoan. Now, if we cannot see these living things with our naked eyes, how can we see them? This is a
Great
question for you all. Yeah, if you want to put that in the chat and somebody
Yes,
just tell me yeah.
I can already see. So, Bhavik, do you want to type it into the chat window? I can see you've raised your hand. Ala says microscopes.
Fantastic. Very well done. That's true. Microscopes. We can see them with a microscope. They can be magnified at 100x or more than 1,000 times, and then we can see actually what they look like. So today we are going to be covering all of these and seeing how do they cause of infection. Now, before we go ahead, the question we ask ourselves, where do we actually find microbes? We can see them in the air, in our mouth, in our food, in our intestinal microbiota, in our tummy, in our guts. You know, your previous session a couple of weeks ago was looking at poop and the gut microbiota, the tummy. And yeah, you have a whole lot of, more than 1,000, thousands of microbes there. They are in our nose, they're in the environment, so they are everywhere, they are around us. We can actually, and this brings me actually to my next slide. And this slide actually is sort of a recap from your poop session because you were actually told, are all microbes, are they harmful, useful, or both? Now I need you to answer this. Again, it's a recap from your previous session, just to see how well you have been following in this session on microbes in general.
This is a great question. So, Bhavik says both.
Okay, any other person wants to answer? Let's just wait
Bhargavi
for
also said both. Ala said both.
Fantastic. Very well done. It's true. More often than not, again, a recap from your poop session, microbes, about 80% or more of them, they're actually good bugs. We need them. They help us. They help our immune system. It's only sometimes when something happens, when there is a bit of disbalance in the system or otherwise known as dysbiosis, this is when they become harmful. So it's good to know that microbes are not bad. We need them to live. We need them to survive. Now, we know that we have these microbes, right, in our system. The question is, how do they get into our bodies? There are different ways by which microbes can get into our bodies. For example, we have the respiratory tract. And in terms of, you may ask, how does it get into my respiratory tract? It's transmitted by air or aerosols. Before I put on the next slide, let me just see how well people can give me, if they have any example at all of a microbe that is being transmitted by air. Again, if you want to put that into the chat, that would be amazing. Let's just see how well we remember those things. Any microbe at all, that can be transmitted by aerosols?
So Bhavik has an answer, which I think everybody's going to say, but it's a really good example. COVID.
Oh, absolutely. Fantastic. Yes, that's why I haven't put COVID in my list. But yes, you're right. Influenza virus, coronavirus, they all get transmitted by air. Also, streptococcus pneumonia also gets transmitted by air. Now, in terms of this is one means of transmission. We also have, again, the entry site is the guts or the tummy, and this can be transmitted by food or water. Now, a bit of a long shot. Does anyone know any example of microbe that can be transmitted through food or water? We can get, any idea? Let's see if anybody has any idea at all.
Wow. So Bhavik already has an answer. He says typhoid.
Oh, fantastic. Salmonella. Well, very well done. Again, that's a very common one. It's caused by salmonella. I haven't put that there. I'm very pleased that Pavik actually said typhoid. He said the disease. You know why? because this leads me to my next question on the slide. Now, in terms of the microbes, influenza virus, what disease will it cause? Do we know what the disease is called?
Great question. So does anybody know what does the influenza virus cause? What's the name of the disease it causes? Allah says the flu.
Fantastic. Well done. This is really good. I'm so pleased we know our microbes and the disease they cause. What about Streptococcus pneumonia? It's a long shot, but there is a clue in the
Yes,
wording itself. Let's see if
yes.
anybody can tell us the disease it causes.
Okay. Everybody's saying pneumonia.
Oh, well, very well done. Very well done. So this is good. If we go also to Vibrio cholera, again, we have a bit of a clue there. Anybody knows the disease it causes?
Cholera.
Cholera. Thank you.
Yes.
Thank you all so much. This is really good. I'm enjoying the session. I hope you will enjoy it as much as I am. So you can see that these different microbes, they have to get into our body in a sort of way. They get transmitted either by the air, the food, or the water. Again, this is why we'll have to be careful as we go along. We're going to talk about why we need those antimicrobial agents, and they can cause us this disease. Now, right? The microbe is now in our body. The question is, how does it cause us infection? So we now have the flu. What is the process of causing this infection? The first thing is exposure. So here, what we have, we have the microbes in red, right? When you have a microbe, the next thing for it to be able to cause the infection, it must adhere. So I'll give a good example with the coronavirus or the flu. They have got receptors. There's receptors they recognize in the human host that enables us to enable the organism to cause infection. If they don't bind to that receptor, they will not cause infection. And this is important when we come to talk about, for example, antiviral agents and how we can stop them from causing us infection. Now, what you can see is the microbe is in red, and then it goes into the body, right? The epithelial cells. Once it does that, the next thing is it invades your system. It colonizes. Once it's colonizing, it's like, I am now the landlord of this body, so I'm just going to multiply and cause the infection. The next thing that happens is the tissue becomes damaged, it leads to disease, and you start having those symptoms. Which brings me nicely to the next slide. How do you know you have an infection? How do you know? What are the signs? What are the symptoms? How do you know that we have an infection? You may have a high body temperature, and so we see this lady that's wearing purple. She's having a rash on the skin. Now I'm going to ask you a question. For the woman that's putting a hand on the head, what do we think is happening to her there? A sign or
Good
symptom of
question.
infection.
Yeah. So what symptom does everybody think that is? Bhargavi says maybe a headache.
Yes, yeah, it could be a headache. And what about the sign of the heart? What will the person be having? What sort of symptom could they be having?
Allah says it could also be a fever, maybe.
Yes, what else could it be? What else?
Okay. Any other? Bhavik says a migraine.
Okay, now let's move on to the heart, on the
Yes.
left panel,
Yes.
just below. Yeah, so what could that be?
Okay. So what could the lungs show us?
Infection, yeah. Let's see if I can
Allah
get.
says maybe a cough.
Yes, yeah, it could be a cough. What else? Yeah, I
Okay.
wonder if people can see my pointer is in red.
Yes.
Yeah,
Yes, we can.
fantastic. Yeah.
Bhavik says asthma.
Oh, yeah, it's those chest signs. Yeah, those are all signs of symptoms to know that the person has infection. Oh, what is happening here now? What is happening?
Okay. What symptom is that?
Do we know what this could be? It's coming from the mouth, right?
Okay. Everybody's saying vomiting. Yes.
Absolutely right. They have a tummy bug, so they're trying to, and then we also have the symptom here, the last one. What could that be?
Okay. Anirudh says maybe a cough again
Yeah.
or
Oh,
sneezing.
it's sneezing. Fun. Oh, well done. Well done, everyone. This is really amazing. So you see what happens. We have them in the air. They go into our bodies. They invade us. They tell us, now you have this infection. You've got to do something about it. The question is, what does our body do when we have the signs and symptoms of infection? This brings us to our immune system. right? So initially, for some illnesses, to be honest, we don't need antimicrobials. Why? Because we have our immune system to fight the infection. So the question is, how do we respond to infection? Now you're having those headache, you have the fever, the cough. So if we have, for example, let's just take bacteria as an example. We have a bacterial infection. Remember what I told you about the receptors? When the bacteria comes into your system, your host, your body says, wait a minute, this is a foreigner. I don't know you. So what do? They do is they try to release inflammatory agents. So they try to fight that infection. They say, okay, you are not part of this body. We don't want you here. And this happens with the innate immune system. It's there to help you fight infection. Usually, the cells known as neutrophils, they come to the rescue. They are the first cells. You see them here. They are trying to invade that bacteria. They are just trying to swallow, like eat them up, because they have to respond to that infection. Now, if our system always responded to infection this way, to be honest, we will not need to take any drugs, right? Sometimes our system become immunocompromised. What do we mean by that? They may become weak either as a result of what we've eaten or other infections that they may not be able to fight this infection. It will really be brilliant if our system works in this way every time. We don't need the drugs, but at other times we don't have a choice, because our system might initially try to fight the infection. They may not be able, and this brings us to antimicrobials. But just before I go into this, let's just see if people have questions so we can take those. But
Yes,
so far, they've done really well, in answering
yes,
my own questions. So it's time to answer their questions.
definitely. So we'll look at some questions in the chat window.
Okay.
But Anirudh had a question, and maybe his question comes from the previous slide where you showed the bacteria invading the host cells, basically. So he
Yeah.
asked, are bacteria living? So, like, are they living when they maybe invade our host cells, when they're inside our tissues? Are they living?
Absolutely. Yes, the bacteria are living. They don't just live. What they do is they multiply to form a quorum. What's a quorum now? They increase. It's like, let's take this example. Let's say one bacteria gets into your system, right? The next thing it does is, you can see here, there were three initially. One goes in and then they begin to multiply. Once they invade, they colonize, they use the human, they use some products of the human host to multiply and form a quorum. So they become so many that it may be very difficult for your immune system to clear it. Yes, they definitely are living when they are in the within the human host.
Right. So they basically have strength in numbers.
That's right. Yes.
All right. Great. So I think we can move on
Okay.
to the antimicrobials, and then we'll come back to questions then.
Okay, then let's go. So, like I was saying, our system may not always be able to help us fight infection, and this is why we need antimicrobials. Ideally, antimicrobials should be used to complement our immune system. It shouldn't be so. I'll give a good example. If you had like the common cold is really common, we get it all the time during the season, right? Rather than just going for an antiviral, it's caused by viruses, by the way. You can try to see, will my immune system, will it fight this infection? Only if it's taking a very long time, it doesn't fight it, before you can say, okay, what else do I need antimicrobials? And remember that for some signs and symptoms, we can actually treat it, with not antimicrobials. Good example, we saw the sign and symptom, the headache. You can take, say, paracetamol for the headache. It also helps with fever. So we don't actually need antimicrobials at that point in time. So we can have antibiotics, which I know everybody has probably heard of. They are one of the things I wanted to emphasize this in this slide, because this is quite common when people are ill, when they have the common cold or the flu. They go to the doctor, they are asking for antibiotics for flu. Just know that antibiotics will not work for your coronavirus. It will not work for the influenza virus. It will not work for the common cold. Any viral infection, it will not work. Why? Because when you had a Mentimeter, somebody said against life. Anti is actually against. Biotics is life. In this case, they are against bacteria agents. So they work for against bacteria agent. Antivirus, from the name, they are against what the viruses Then we have the antifungals against fungi like the candida, the ringworm. We will go into that in more details when we come to the agent itself. Antiprotozoans actually is a very, it's like a two group, so to speak. We have got the antiparasitic agent. Aha, let's just see. There's a very common one, which is common where I grew up from and probably common in India too. And let's just see if people can tell me an example of a parasite. You know, an example of a parasite. And I'm not going to give you any more clue to that. Let's just see.
Okay. Right. Let's test. Okay, Bhavik says tapeworms.
Yes, that is an anti-helmintic agent. It's a helmint. Yes. What else? That's really common. Kills
Okay.
so many children
Ala
and
says
adults.
malaria.
Malaria, thank you. Brilliant. It's fantastic. And why did I say this? Because when I come to my anti-protozoan slide, malaria is what I have used as an example. So when our system, when they can fight all of this infection, we have to take one of these. Either we take antibiotics for bacterial infection, antivirus for viruses, antifungal for fungal infection, antiprotozoan for things like tapeworm or Plasmodium falciparum that causes malaria. Now,
So,
let's just
Blessing,
do,
maybe just
yes,
to really drive home this message again. For every different category of microorganisms, there's a different antimicrobial.
that's
You
right.
can't use the same one. You can't have a medicine that works against both bacteria and viruses and fungi.
No, usually you have an antibiotic, it works mostly for bacteria, antivirus work mostly for viruses, antifungi mostly for fungi agents, antiprotozoan. There are very few exceptions. There is a drug called metronidazole. It is used, it's an antibacterial agent. It's used against C. diff infection, Clostridium difficile, that can cause some tummy infections. But also that drug is also used for parasitic, sometimes used for parasitic infection. But we do not have so many drugs like that where they are used for both bacteria, viruses, fungi. I haven't used, one drug that's used for everything. Sometimes the drug might be modified for a different cause, but more often than not, they are setting for that same class of organism, like either they are bacteria or viruses, fungi, or protozoan. And this is why
So,
it's not good to go just off the shelf,
exactly.
and get,
So
so
when
you
you get
need
an
to get, yeah,
infection, you need to know what is causing the infection.
exactly. So you need to go to the doctor, they need to carry out tests and say, okay, is it a bacteria? Is it virus? Is it fungi? And then you can be given the appropriate antimicrobial agents.
Okay, so Anirudh has a really, really nice question.
Yes.
Yes, if we take antibiotics, do antibiotics also kill the bacteria that are helpful to us?
Oh, it's brilliant. He's gone way ahead of me. I will answer the question now, but you're going to see that when I come to antibiotic resistance, the answer is yes. Because when we come there, you're going to see that we have different classes of antimicrobial agent, broad spectrum, narrow spectrum. Unfortunately, yes, they will also reduce the amount of good bugs we have in our system. And this is the reason why we should only use them when we really need them and not just use them because, we think it's going to help our infection. That's a very fantastic question, by the way.
Great. I think that was really good. And then, so let's move on. So then we'll come back to answering this when we go to the antibiotic slide.
Okay. So, yeah, actually, this is now a question for them because we have been doing a bit of talking and just say, so they just need to put the answers right on the chat. So when we say an antibiotic, I know this is really easy, but let's just recap. What will be the answer to this question? I should have done A. Oh, let's call it A, B, C, D. So A is influenza, B is, C, then D is for drugs. So is it A, B, C, or D?
Okay, which one is what is an antibiotic?
Yeah, what is an antibiotic?
Okay, I can see Bhavik has said D. Anirudh has also said D. Bhargavi has said D. Everybody's saying D.
Everybody said D. Fantastic. That's amazing. You're right. There are drugs that are used to treat bacterial infection. Now, let's talk about bacteria itself. What is bacteria here? So we are out with D. We are left with A, B, or C now. So this is A, if it helps, B, and C.
So Bhargavi says C.
That's right. Yes, a very small microbe that can be helpful or harmful. Now, which of these can be transmitted by air? Remember our first or second slide when we're looking at transmission of microorganisms?
Yes. Okay. Bhargavi and Bhavik both say A.
Okay, now the last answer to the last question is quite easy. It's just there,
Yeah.
right? Symptom, we did say it's a sign of disease of illness, can be headache, fever, cough, So these are just terms that we needed to remind ourselves on. Now let's come on to look at antimicrobial agents themselves. So I've started with antivirus and I've looked at antibiotics last because I know most people already know that, but I also want to talk about antibiotic resistance at the end. So antivirus, they are used against viruses. Before we take a look at the antivirus, right, we need to know how they will act. And this brings us to the viral replication cycle. Remember what I told you initially? For most organisms to cause disease, they need receptors. They must attach to the human cell host. If they are not able to attach, they can cause us infection. So we'll quickly take a look at this. So we have the virus, right? It's interacts or gets into the cell with this receptor. Once it does that, it goes right into the cell, which is otherwise known as endocytosis, and it will uncoat itself. It's like taking its coat, bringing out all its genetic material, the DNA, the RNA. Why is it going to do that? Which brings me to the question someone asked previously with bacteria. Will they multiply in the host? Yes, viruses have to do this. Actually, viruses cannot replicate without the human host. They need us to replicate. They must do it within the host. Do you know why? Because they do not have the enzymes to do that replication process. They need that human host. So this is why they always have to infect us so they can grow in numbers. So once it has taken out its DNA, its RNA, it will replicate or multiply. After doing that and it's happy, now it has got small mini viruses, it's going to now assemble itself again and then get out of the human cell to infect the next host. The good news is at each of these time points, there are antiviral agents that can be used to deal with it. So if any of this, it's like a sequence. If one of the sequence is missing, then the virus will not be able to cause infection, which is why in terms of antiviral agents, we have got things known as entry inhibitors. So what do they do? So a good example of this is it's called docosanol. It's they will not allow this virus. So this is why we have all of these signs. These are the plates where the different points whereby you can actually stop the virus from causing infection. They will not allow the virus to bind to the cell. If it doesn't bind to the cell, it cannot go into the cell. It cannot replicate. It cannot cause infection. We also have another group of virus. Now, you know what? This virus is very smart, right? It's gone into your cell. It needs to uncoat itself. Before it gets to that stage, if you give the person inhibitors of viral coating, antivirus, the virus again is unable to uncoat itself, and what happens? It cannot release its genetic materials. And a good example is a drug called amatadine. This is sometimes used to treat the influenza flu virus. So its mechanism of action is by inhibiting that virus from uncoating. Next, remember what I said. The virus has got into the cell, it was able to uncoat itself, but now it needs us to be able to replicate because it needs those enzymes. So now some virus, they inhibit the polymerase, so they will not allow that reaction to take place. That's why they're called polymerase and protease inhibitors. So it cannot get transcribed or into RNA or DNA, or translated rather into proteins. So again, if the virus had been able to do these three steps, when it comes here, it's been blocked by these inhibitors. And this is really, really very important because, for example, during the COVID, I have the drug at the end, the remdesivir that was being used for coronavirus. It works as one of these inhibiting the DNA replication so that it cannot cause infection. And then finally, we have those that will inhibit viral release. We assume the virus has been successful, but now we don't want it to go out and infect other people. So example of this is the newer drug now used to treat flu known as oseltamivir. And what it does, it stops the virus from releasing from that cell. So you can see for each of the life cycle of the viruses, there are antiviral agents that are helpful to make sure that that process is not complete to cause some infection. Do you want us to take question or move on to the next?
So I think we can move ahead
Okay.
and we'll stop after the antifungals for some questions.
Fantastic. Now let's move on to the antifungals from the word against fungi. Just like the viruses, for us to understand the mechanism of action by which these act, we need to understand the cell structure. So the fungi have got a cell wall that contains beta glucan. Glycan are like sugar units. It's got a cell membrane. This is really important, that contains ergosterol. Do you know why? You may have heard the term cholesterol in humans. Humans have cholesterol. Fungi have ergosterol. And this is really important. You know why? Because some antifungal agents, they are directed towards ergosterol. It means, unlike antibiotics, like back to the question I was being asked, if it's going to kill the good and the bad bugs, which is right. In this case, the antifungal agent, because it's directed at ergosterol, it's not going to have an impact on the human cells. So we can see, for example, this is just a fungal growing on a plate. Out of interest, I have put this image. Do we know what this could be? I've sort of zoomed in. What sort of infection is this caused by fungi? Anybody knows? Somebody will probably know. Let's see.
Okay. Anirudh says it looks like dandruff.
Yeah, he is very close. He is very close. What else? What's the other disease? What's it called? It's very, very close. Yes. Any other person know the name of the disease itself? Or what causes it?
Okay. Do we have any guesses on what the disease could be? What could cause it?
Any of them?
Nope, I don't.
Okay, but
Okay,
he was.
Bhavik is asking, is it a tonsil infection?
It is, but what this is, somebody said dandruff, and that person was really close. Remember the ringworm? It's not totally clear. So fungal agents cause like ringworm infection, and when that happens, some of the skin, remember the hair on the skin just goes out. This is why the hair is this way. And here, what we have is, I don't know if people are going to know what this is. If it helps, I'm going to describe it because that picture does not show some of those plaques. They are like white plaques on the mouth. That's also is caused by fungi. So it's caused by candida infection. Now, in terms of the drugs that might be used to treat this infection, so for something like this infection on the right, the candida, we have the flucytosine. It will inhibit DNA synthesis. So one of the things you're going to notice across all of these antimicrobial agents is it targets the different stages of their life cycle. So it's either inhibited the DNA, the cell wall, or the cell membrane. And here we have this group, the azoles. They inhibit membrane, cell membrane. And azoles are a group of antifungals, for example, we can use to treat this ringworm infection. So these are some of the things that we need to know. Whereas some other antifungi will disrupt the cell membrane. So if any of this life cycle is disrupted, it means that that infection is going to be stopped. So let's take some questions at this point in time.
Yes. Okay. So one of the questions is, why does our body, Anirudh, why does our body release cholesterol if it is considered bad for us?
It's a very good question. Remember that we have the HDLA and LDH. Not all cholesterol is bad. Some of those cholesterol, what are they? They're fatty substances. We actually need it for our body metabolism. It's just when you have a high concentration, that's when it's usually bad. And for most things, even like sugar, when it's too high, it becomes bad. But we need some of these elements in our system for it to function properly. So not all cholesterol is bad. There is the good cholesterol and the bad cholesterol. That's a very good question.
Great question. Yes. So another question is, so based on what you said with antifungals as well as with antivirals, it looks like we have multiple types of antifungals and multiple types of antivirals, and all of them affect different parts of the cell or different parts of the way that the microbe infects us.
That's right. Yes.
So are there certain types of medicines for certain diseases, or can you give any antifungal for any fungal disease?
It's a very good question. No, you cannot again give any antifungal for anything because one of the things is to understand the fungal infection and its mode of action, and then the antifungal agent will be targeted towards that. You understand? For some illnesses, you can use multiple antifungal. So there are some you can use amphotericin B and the azoles for, whereas others it has to be very specific. It's similar to like bacterial infections and even the viral infection, depending on the mechanism. And mind you, this is based on years of research that has been done in the lab to these organisms to know what is the best antimicrobial agents to use.
Exactly. So scientists, doctors, all of them need to know what is causing your infection.
Yes,
Is it a virus? Is it a fungus? And they also need to know how does that fungus affect your body.
exactly. If they don't have that information, it's just going to be a bit of guess, and that's not what we want because that will lead to antimicrobial resistance.
Yes, definitely. All right. I think we can move ahead. And then
Okay.
we'll stop again for questions after the next two sections.
Okay, now let's look at antiprotozoans. And again, so, somebody hinted this at the beginning, talking about tapeworm and malaria, which I thought was really good. Just like the antiviral agent and the antifungal agent, we also need to know the life cycle for us. It's just what you've just asked me now. So this brings us nicely into this. Yeah, we are going to be looking at antiparasitic agent that affects malaria. So this is, remember, it's caused by Plasmodium falciparum. What happens? The mosquito takes a bite, it takes a blood meal, a delicious blood meal from the human host, and it releases something called the sporozoites. These are green. When it happens, the site of that infection is in the liver. It goes to the liver. And here, this is the image of the liver. The liver cells become infected. Now, in terms of their stages, again, from the liver, what happens? The sporozoites, they will become mature. So they grow in the liver. They mature to something called schizons. Now, when those schizons, when they are mature, do you know what happened? This is schizons you have here. So the sporozoite here, the next thing, they are not mature, they will rupture, they will open. And this is why you now see them like in red, right? So it's a different stage of the life cycle of that of Plasmodium falciparum. So I'll just call it for malaria, so it's easy, which is the disease it causes. Now, depending on the stage, so if it was in the infected liver cell, we have got the primaquine that can be used to treat that infection. Now, what else can we use to treat the infection? At the red blood cells. So where does it get to the red blood cells? Remember, when it's injected into the human host, it's a sporozoite, goes into the liver, matures, and then opens up and then becomes a merozoite. Now, the merozoites will now migrate to the red blood cells. And that's why you always hear, they are called erythrocytes. So when they come into the red blood cells, we have got this powerful antimicrobial agent. These days, some species have become resistant to it. So sometimes it's being used in combination. So we have got the artemisinin, and this artemisinin actually is gotten from sweet worm wound that can kill Plasmodium falciparum. When it's at this RBCs for red blood cells, when they've become merozoites and go into those red blood cells to infect the red blood cells, if you take this artemisinin, then you are likely to, the illness is likely to be resolved. Now, the malaria is one of, or Plasmodium falciparum has got two life cycle, the asexual and sexual stages. And the asexual life cycle, do you know what it does? So that's the red blood cell. It will now differentiate to become a gametocyte, then starts the whole process. So it's like a cycle. At each of these different stages, we have the drug. You may have had chloroquine. I had that when I was a kid. And so
So
against
basically,
malaria
even
infection.
here, maybe just to simplify it for them, even here at different stages of how the parasite affects our body, there are different
Yes.
medicines.
Absolutely, absolutely. So it's again good to know. We don't have that information, but doctors will usually have it because they do blood tests and they can tell, in the lab at what stage this infection is. So again, each of these anti-parasitic agents in this case is they may affect the DNA replication. For example, the primaquine, the chloroquine or the quinines, they will affect the DNA from replicating. So we can see that depending, again, just like we have for, it is really, if we want to prevent antimicrobial resistance, it's very important for us to know, not us, the doctors per se, to know what stage of infection we are in so they can treat us with the appropriate antimicrobials. Now, this brings us to the antibiotic agents. Remember, the antibiotics are for bacteria only. One more time, we can use them for viruses, we can use them for fungi or antiparasitic agent. With the antimicrobials, we have things known as the broad spectrum. So when we say broad spectrum, this is why I've put quite a lot of bugs here. They can kill so many things, so many organisms. And narrow spectrum, they can only kill few. They are very specific for the organism they kill. And somebody said, somebody mentioned penicillin when we did the Mentimeter. It's an example of a narrow spectrum antibiotic. And again, it has to mechanism, we'll come to that in a moment, has to do with the cell wall. The other thing that we just need to know about is not all antibiotics will kill the organism. Some will kill them, and these are described as being bactericidal. Others are bacteriostatic. What does that mean? They will stop the growing. Let's just quickly take a look at this graph. Here, what we have is the number of viable living bacteria. So the bacteria is growing here. When you add the antibiotic drug to it, if it's a bactericidal drug, what happens is the microbe is just going to die with time. However, if it's a bacteriostatic drug, what happens is it's just going to inhibit it from growing. Once
So
you take away
static, it doesn't grow,
static,
it doesn't
exactly.
die.
Yeah, exactly. That's an easy one. Once you take the drug away, it can go again, it can grow. So you can see that again, depending on the infection as well. And I think the question may be, why don't we just give them bactericidal drug?
Yeah. Why don't we
Yeah.
just kill everything?
Why don't we just kill everything? I think it's gonna depend on the organism. Remember, let's just go back. Why are we using antimicrobials in the first place? Because our immune system are not able to fight the infection. If the doctor gives a bacteriostatic agent to suppress the bacteria, it may suppress it at a point that your immune system can come to the rescue after that bacteria has sort of stopped growing. It's not dead, but here what happens is it's called stationary phase in the life cycle of a bacteria. It's not dead, but at this point, your immune system might just be able to fight the infection. And so the doctor will say, okay, let's take this agent for 7 days and see what happens. And if after that you don't get better, he may decide, okay, let's change it, or let's see how the immune system have responded to infection. So it's really important that we know the differences between these terms, because when a doctor gives us a drug, it's, it may be that he's decided to give us a bacteriostatic agent because for that organism, it's not a virulent, or it doesn't cause so much disease. It's not going to kill you. It's not going to make you die almost immediately. But if whenever the risk is high, that is, is a drug, for example, a bacteria that can lead to death quickly, then it's likely the doctor will give you a bactericidal agent. So it's good to sort of know the differences between these two.
So now, maybe just one question before we move ahead.
Yes, please.
Really nice question in the chat window. So does that mean that if we were to give bacteriostatic antibiotics,
yes,
versus bactericidal, does this change the kind of memory that our immune system creates of the infection or of the bug? Does bacteriostatic help in creating more memory cells in our immune system?
To an extent, I would say yes, because that bacteria has been in the system recognizes it, and hopefully is able to fight that infection. So yes, if it's encounters the bacteria another time, it should be able to respond faster to that infection. Yes, it does create that, because the memory is long-lived compared to if you give a bactericidal agent and it kills it almost immediately. It's a very good question.
Great. Thank you so much. Let's move on, and then we'll come back to it I think in the interest of time, we'll finish this section now.
Okay.
Come to question.
Now, let's take a look at the antibiotics mechanism of action. Remember the antifungal agent? We had a similar slide, right? We have the cell wall. So bacteria have got cell wall made of peptidoglycan. And actually in the lab, if you're working in the lab, you can actually stain the cell wall. Some of them are known as gram-negative, others known as gram-positive. The question is, where did this name come from? It came from a scientist, Christian Gram, who first did the experiment in the lab to show that the peptidoglycan layer of the cell wall is thick. If it's very, very thick, it's gram-positive. If it's thin, cell wall is gram-negative. So this is how they came into place. So some antimicrobial, for example, penicillin, that was the first antimicrobial discovered, they will inhibit that cell wall from forming. If the cell wall doesn't form, then the bacteria cannot do anything. It cannot infect, it cannot process, it cannot cause you any infection. Others will disrupt the cell membrane. As usual, for all of these organisms, inhibiting DNA or RNA synthesis is just like the bacteria is all there. It cannot replicate itself. And then we also have those that will inhibit protein synthesis. A very good example I want to point out here is the aminoglycosides, because current research on this, which we'll see in the later slide, has seen that some of these antibiotic, when you heat up the bacteria and add some of these antimicrobial agent, it actually kills some of these organisms like the gram-negatives faster. Why is this so? We come to mechanism of resistance. So just like the antibacterial, antiviral, antifungal agent, antibiotics have also their mechanism of action. It may be to inhibit the cell wall or to disrupt the cell membrane or DNA or RNA synthesis.
So maybe one quick question,
Yes,
which
let's go
again I
for it.
think is a great question. So
Okay.
I mean, even our cells have cell membranes, or do if other cells around the host cell also has these components, will the antibiotics also affect our host cell? I mean, cell membranes or cell walls?
No, because we don't have peptidoglycan. This is restricted to bacteria only. So this is why you remember, just like we said for the antifungal, the ergosterol and the cholesterol, our cell membranes are different from what the bacterial cell membrane has. So antibiotic agents will not have an effect on our cell membrane. It only works against the bacteria infection.
Great. Thank you, Blessing.
Okay, let's move on to the next. Oh yeah, I wanted to show you the slide because obviously, I used to be in the lab, and Snena, I'm sure you probably have done this at some point too. It's really great. The question you may ask is, how do we know the antimicrobial is working? How do we know the antibiotic is working? So in the lab, we'll usually grow bacteria in like an artificial media in an agar plate. What you cannot do, remember those antibiotics we just talked about, the penicillin, the vancomycin? Each of them, you can put them on the plate, spread the bacteria on the plate. The next thing you want to find out is the bacteria. Why are we doing this actually? We want to know, is it sensitive to that antibiotic or is it resistant? So you grow this up in the lab for about a day, overnight. The next morning, when you come, what do you want to have a look at? This is your antimicrobial disc. This is your bacterial growth. This is your zone of inhibition. If the antimicrobial agent, for example, penicillin, was able to inhibit this bacterial growth, we said is what? Sensitive. On the other hand, if it was able to resist it, we say is resistance. Now, I just want to be sure that, and this method is called disc diffusion method. Let's just see if my explanation is clear enough. So I have an actual plate. The question now is between A and B, is A sensitive or resistant? They will need to answer this for me. Let's
Great,
just see
great
if they
question.
have understood that.
Yes. Okay. So look at A and see where is the disc, where is the bacteria growing, is there a zone of inhibition around the disc? And tell us, if you saw this plate, if you were a clinician and you saw this plate, what would you say? Would you say it's resistant or sensitive for A, just for A? All right, we'll wait for a
If you just
few
type your
people to,
answers
yes, you can just
in
type
the
it into
chat.
the chat. Okay, Bhavik says resistant.
Okay, let's just wait for one or two more persons to
Yes,
answer and just see their following.
yes. Okay, Pranav says it would be sensitive.
Okay.
Okay, do we have anybody else who wants to add in what they think? Anirudh says resistant.
Okay, let's just explain that. Oh, now for B, so B will be the
Yeah.
opposite. So I'm not going to. So when we say a bacteria here is sensitive, there has to be a zone of inhibition. And just to confirm that those who said resistance is right, because there's no zone of inhibition here, what does it mean? It means that antibiotic was not able to even suppress or kill the growth of that bacteria. So it's resistant to it. And this is quite important because we're going to come to antibiotic resistance in a moment. Whereas in B, we see the zone of inhibition. It means that the antibiotic or the bacteria is sensitive to B because it was able to inhibit its growth. Now, let's just take a look at antibiotic usage. They are having a lunch, right? During lunch, Eva was talking to her friend Anne about her headache and runny nose. So she's giving her the symptoms. She has headache and runny nose. What does she say? It really hurts, and I think I'm getting a cold. This is very common if you're getting a cold. Don't you have any antibiotics at home you can take? And ask her. What did Eva say? That's a good idea. We still have some from when my sister had an ear infection. I'll ask my mom. Do we think it's actually a good idea for Eva to take her sister antibiotics? Yes or no?
Okay, everybody's saying no in the chat window. Big no.
Fantastic. Very big no. It's not, because it is not, it wasn't prescribed for her. And also headache and runny nose. It's very possible she's actually getting a flu or a common cold. Antibiotics will not even work for that. But however, even if it was a bacterial infection, because it wasn't prescribed for Eva, she cannot have that antibiotic. This brings us to the mechanism as to how antibiotic resistance develop. Now remember, there are a lot of germs, but few are resistant. If we take a look at this picture, we have the blue and the orange. So the blue are the good bugs, the red are like the bad bugs. When you take an antibiotic, which brings me to an earlier question somebody asked, if the good bacteria will also be killed, which I said yes, it's true. So they will kill the bacteria causing illness. They will also kill good bacteria that is protecting the body from infection. Now, sometimes not all the bacteria will die. What do? The antibacterial resistant bacteria, they will multiply and cause infection. Do you know what these bacteria can do? They've got the genes that actually, some other time, they might actually pass some of those antibiotic resistant genes to other bacteria in your system to cause you more trouble. And this is why it's really, really important that when it comes to taking antibiotics, they must be prescribed by the doctor. We must finish the course of that antibiotics. Now, let's quickly take a look at the mechanism of antibiotic resistance, and maybe we'll take a question after this. We have,
in the interest of time, we can just explain this broadly, and then we'll come to a few questions.
okay,
Yeah.
so we have the influx and efflux of antibiotics. What does this mean? Let's take a look at this quickly. We see this organism, it needs to go into the cell. We have the porins. So porins allows molecules to go into the cell. If the bacteria is resistant, what happens is it will not allow that antibiotic to go into the cell. So the antibiotic is in red. And some bacteria, they've developed very special mechanisms to sort of pump out. It's called efflux pump. They've got efflux pump to take out the bacteria, antibiotic out of the cell. So what does this happen? Because we have resistant bacteria, they will decrease the amount of antibiotic going into the cell, but they will increase the amount of antibiotic going out of the cell. Why do they do this? Because they have the genes that enable them to do this. At other times, some antibiotics become modified by that particular bacteria. Instead of binding to a particular target, the bacteria has got enzymes that will degrade that from happening. If they cannot bind to the target, what happens? They cannot fight that infection. Finally, at other times, we have antibiotics that can modify the antibiotic targets, right? So we have the antibiotic, it cannot bind to the target. Why is it doing so? It's called target protection. It changes it. How does all this happen? Genetic mutation in the gene for that particular incident, the bacteria has it, it changes the sequence, and the antibiotic is not able to bind to infection. A good example of that is in vancomycin that is normally used to treat Staphylococcus aureus infections. Do you want us to take questions now?
Yes. So we
Let's
do
go
have a
for it.
question in the chat window from Bhargavi.
Okay.
She asks if antibiotics can kill both good and bad bacteria. So then how do some of the bacteria actually survive when you take antibiotics?
It's a very good question. It is because of the genes they have. Sometimes, if people take antibiotics for that infection, do you know what you do? You're not doing your system, your immune system, a good thing, because the organisms have been taking, you've been taking antibiotic for a particular infection. Sorry, not for a particular, let's say for viruses. You didn't have an infection. You were just decreasing the amount of good bugs you have there. What happens is the amount of bad bugs increases. The actual time when you now have a particular infection that is supposed to kill the bad bug, it won't kill it. Why? Because it's developed that resistant mechanism. It now has genes that enable, so it may be by any of this mechanism I've just mentioned. It now has those genes that will not allow it for the antibiotic to function properly. And that is why it is called antibiotic resistance. So that's what happens in that case.
A, Another good follow-up question is, then can we just do something to try and kill only the bad bacteria? Do we have medicines that do that?
Absolutely. These days, they are beginning to like generate medicines that are narrow spectrum, so they can only target a couple of bacteria and not others. Also know that, for example, some antibiotics, they work towards some certain organisms. So instead of giving broad spectrum, narrow spectrum antibiotics are now being used. And also there is this development of trying to use more like natural ingredients towards bacterial infection instead of using broad spectrum antibiotics. So that would really, really help. Which is why, somebody mentioned, and I think I need to sort of highlight on that. When we had a Mentimeter, somebody talked about bacteriophages. That was really, bacteriophages are what? Bacterial viruses. They are viruses that actually eat up bacteria. That's also another research going on. They will not affect the human host, but what they do is to actually eat up bacteria that cause infection. So these are all new researches that are going on. They haven't been licensed yet, but it's nice to know that we have this sort of research going on for against bacterial infections.
Yes, definitely. So there is hope of different ways of treating these infections.
That's right. Yeah.
Okay, I think since we are close to wrapping up, maybe we'll just finish the last few slides and then we'll go to taking our post-session feedback and polls.
Okay, let's quickly do this then. Now, the question is, what causes antibiotic resistance? And is really, this is a slide from the World Health Organization. Over prescribing antibiotics, for example, when somebody has a cold, they give them antibiotics. That's not good. Patients not finishing their treatment. You have a disease and the doctor tells you, take the antibiotic for 7 days. After 4 days, you feel great. You're like, no, I'm not going to take these antibiotics again. Please do not do that. It's really important to finish that course of antibiotics. And sometimes, antibiotics are used during famine. Again, this can be transmitted to humans. And also poor infection control in hospital and clinic. We also have the lack of hygiene and proper hand sanitation. Remember the Happy Birthday song for washing of the hands? Also, because of all of this, we also need for the development of new antibiotics. This I saw from last month or so from the conversation that antibiotics are being inappropriately prescribed for COVID, increasing the threat of antimicrobial resistance. Just remember, they do not work for viral infections. So quickly, we have a minute. Let's check your knowledge. Antibiotics can be used to treat viral infections. Yes or no?
All right, let's see what everybody says. Okay, everybody, I think I saw lots of no's in the chat window.
Fantastic. Most of the time, the immune system can fight infections like the common cold by itself. Yes or no? True or false?
Yes, lots of yeses.
Okay, finally, antibiotics kill both good and bad bacteria. We've said it before, so that's true.
Yes.
And quickly, if we take a look at current research on antimicrobials, these were published recently. Heat up antibiotics. Remember when I talked about the aminoglycosides? When they heated E. coli, it's a gram-negative bacteria, which is usually difficult to kill, and added the aminoglycoside, what happened is it became effective and it was able to kill those bacterial infection. And sometimes, again, in developing countries and even developed countries, if antibiotic is given to a pregnant woman just about to give birth, that will reduce sepsis. Sepsis can be a very fatal infection during childbirth. And during the COVID season, we also had 2 antiviral agents that were sort of approved. One is an immunomodulatory event, so modulates the immune system, whereas the other is an antiviral agent. So these are so just all hope is not lost. There are still so many, scientists, pharmaceutical companies that are doing research on different antimicrobials so that, we can live in a healthy environment. So Quickly, let's summarize because I think I've come to the final slide. Remember to only use antibiotics that are prescribed to you by a doctor. Very, very important to finish the course once you have started it. Antibiotics do not work for viral infection. Proper hand washing with soap and water for 20 seconds. Remember the happy birthday song? Happy birthday to you. If you try the song, sing it for 20 seconds. You're going to wash your hand really, really properly. So thank you so much, everyone, for your attention. It's been a pleasure. And yeah, if there are any questions or if you want to take them in your poll, that's absolutely fine. Thank you.
Thank you so much, Blessing. That's a really nice way to remember how long to wash your hands.